Hey there, future biomedical engineers. Get ready to explore the fascinating world of microbes. These tiny organisms are everywhere, from the air we breathe to the food we eat. We're talking bacteria, fungi, viruses, the whole microscopic gang. Now you might be wondering, why should we care about these little guys? Well, microbes play a huge role in our lives, both good and bad. Some microbes cause diseases, while others help us digest food, make medicines, and even clean up the environment. To understand these microscopic wonders, we need to grow them in the lab. That's where microbial cultures come in. It's like gardening, but instead of plants, we're growing tiny organisms that we can't even see with the naked eye. So, put on your lab coats, grab your petri dishes, and let's dive into the world of microbial cultures. So, what exactly is a microbial culture? Simply put, it's a method of multiplying microorganisms in a controlled laboratory environment. We provide them with food, water, and the right temperature, and they reward us by multiplying and forming colonies that we can see and study. Think of it like this. You wouldn't try to study a lion in the wild without observing it in a controlled environment first, right? The same goes for microbes. Microbial cultures allow us to observe their growth patterns, identify different types of microbes, and even test the effectiveness of antibiotics. Microbial cultures are essential in various fields, including medicine, research, and even industry. Doctors use cultures to diagnose infections, while scientists use them to develop new drugs and vaccines. Pretty cool, right? Now let's talk about the different types of food we can give our microscopic friends. Just like us, microbes need the right kind of food to thrive. That's where culture media come in. These are specially prepared mixtures of nutrients that provide everything microbes need to grow. One common type of culture media is nutrient agar. It's like a delicious soup broth that's been solidified with agar, a jelly-like substance. Nutrient agar is great for growing a wide variety of microbes, but sometimes we want to be picky eaters and only grow specific types of microbes. That's where selective media come in. These media contain ingredients that inhibit the growth of unwanted microbes while allowing our desired microbes to flourish. And if we want to be extra fancy, we can use differential media. These media contain indicators that change color in response to specific metabolic activities of microbes, helping us differentiate between different types. Think of it like this. Nutrient agar is like a buffet open to all microbes. Selective media is like a restaurant that only serves Italian food, attracting Italian food lovers. And differential media is like a restaurant with color-coded menus, making it easy to spot your favorite dishes. Now that we know how to feed our microbes, let's talk about how to grow them in a way that allows us to study them properly. That's where streaking techniques come in. Imagine you have a mixed bag of M&Ms, and you want to separate them by color. That's kind of what we're doing with streaking techniques. We're trying to isolate single colonies of microbes from a mixed population. One common streaking technique is the streak plate method. It involves using a sterile loop to spread a small amount of microbial culture over the surface of an agar plate in a specific pattern. This helps to dilute the microbes as we streak, eventually resulting in the separation of individual cells that grow into isolated colonies. Other streaking techniques include the spread plate method, where we spread a diluted sample of microbes evenly over the surface of an agar plate, and the pour plate method, where we mix a diluted sample of microbes with molten agar and pour it into a petri dish. Each streaking technique has its own advantages and disadvantages, and the choice of technique depends on the specific experiment we're conducting. But no matter which technique we use, the goal is always the same: to isolate single colonies of microbes for further study.